Follow the reaction arrows. Follow the reaction arrows. Follow the carbon as it goes. Okay, so all jokes and bouncing aside, metabolic pathways, the best way to understand them is to really just choose molecule and track it. Follow those arrows. It can be really overwhelming to look at the metabolism in its full, beautiful entirety. This isn't all of metabolism, but even just looking at kind of this core metabolism, it can look really overwhelming. But because the beauty of metabolism is that everything is interconnected, but that interconnected nature of everything, those molecules being able to go back and forth between different pathways depending on what enzyme you use, and then having the same enzyme used in multiple pathways, whew, there's a lot to keep track of. But thankfully, you can just track things. The way that you track things is, well, just pick a molecule and follow it. In metabolic tracking studies, people actually use like radioactivity to track molecules as they go throughout different pathways, they go throughout the body. But then once they figure all that out, they make these nice charts that we can look at. And now we can follow those molecule states without having to kind of go and work in a thought room where we work with radioactivity and have to have Geiger counters, fancy stuff. The way that metabolic pathways are typically drawn out tell you a lot about how those pathways work and operate. And they tell you enough that you can really follow along and think intuitively and critically about when different enzymes might want to, when you might want to use different pathways and things like this. There are some key things that you want to focus on when you track molecules as they go throughout a pathway. One is going to be the directionality of arrows, whether or not a reaction is reversible, as indicated by kind of like these bidirectional arrows, or whether a reaction is metabolic irreversible, which we represent with these single directional arrows. You also want to track any cofactors that are coming or going, which are going to be indicated by smaller arrows, kind of curving away from the big arrow, showing you what goes in and what goes out. It's kind of here that you're going to see important things like our reducing agents. Are we regenerating our NADPH, our NAD plus, things like that, as well as when we are using energy. So like our ATP or other NTP equivalents, and when we're kind of like gaining it. So keep your eye on kind of what's coming and what's going apart from the main carbon skeletons that are going to get the most of the attention. So look at those things that are coming and going above the arrows. Look at the directionality of the arrows, and then look at kind of the regulatory features that are often going to be able to be shown in different ways. So in my charts, what I've done is I have like these green arrows that are indicating that something is going to be activating, and then a red X is something that inhibits. So, for example, glucose 6 phosphate dehydrogenase is going to be activated by NAD plus, and it's going to be inhibited by NADH. I bring up this example because it's a really great example of how we can use the information both about the regulation and about when we're generating what in order to kind of think through when a molecule might want to go different phase. For example, let's start and say that we had glucose and it comes into the cell. We can see that for each of these enzymes, we have a one-directional arrow. That's indicating that these reactions are metabolically irreversible. Remember that nothing is technically irreversible, but due to either an inherently large negative delta G naught, so basically they're inherently really energetically favorable in the direction as written, or basically because of really skewed concentration, so you have a lot more of your reactants in your product, or some combination of both, the reaction is not going to be reversed under physiological conditions. So, for example, our hexokinase step, if we want to reverse it, we actually need to use a different enzyme, this glucose 6 phosphate. We can see that this hexokinase is going to then phosphorylate glucose to give us glucose 6 phosphate. And now if we look at the glucose 6 phosphate, we can see that it's going to go a variety of different ways. Some of these ways are going to be easily reversible, such as this phosphoglucose isomerase step and this phosphoglucose matrix step. Note that when we have isomerase reactions or we have mutase reactions where we're kind of just shifting things around, those are often going easily reversible and have like a low energy barrier. However, we can see that if we want to go in this pentose phosphate pathway, well, here we have a one directional arrow at that glucose 6 phosphate dehydrogenase step. That's going to tell us that this is kind of like an irreversible step. When we have these irreversible steps, these are typically the steps that we're going to want to highly regulate because, well, we can't easily go back. So now we have to think about, okay, well, why would we want to go down glucose 6 phosphate, use glucose 6 phosphate dehydrogenase? Why would we want to use the pentose phosphate pathway? To understand this, even without knowing what the pentose phosphate pathway even is, with barely knowing anything about it, we can just look and see what it's going to generate. We take this glucose 6 phosphate and we can see that in this first step, we're generating NADPH. And then later we generate another NADPH. Well, what do we need NADPH for? What's it good for? If we look at our pathway, we can see, well, where do we else do we see NADPH metabolism? Oh, we see that it's going to be used to make our fatty acids, to make our lipid. So if we want to make lipids, we're going to need NADH. And well, where do we make NADH? We can make it with the pentose phosphate pathway. So maybe we want to go this way if we want to make lipids. We can also see that we can use this pathway to generate DNA and RNA and coenzymes. So maybe if we want those things, we're going to want to go this way as well. So we can already start to kind of see, okay, well, maybe if we want, we have rapidly dividing cells, we need to replicate our DNA, we need to make lipids to make more membranes. If we have these things going on, then we'll want to go the pentose phosphate pathway. Again, we didn't even know anything about the pentose phosphate pathway going into this, but we can already tell. But how are our cells going to know that? Well, they're going to know based on what is available or what hormones are being used. And that information is then going to be given to us in the form of the charts telling us what it's activated by or what it's activated by. So if we look at that first step, that glucose 6 phosphate dehydrogenase, we can see that it's activated by NADP plus and inhibited by NADPH. Well, this makes sense. We said that this pathway can generate NADPH. So if you already have a lot of NADPH, then you wouldn't need to do that. So you'd want to inhibit it. But if you don't have a lot of NADPH, well, that's because most of it's in the NAD plus form. Because remember, those are just kind of like going back and forth between the same thing, whether or not they have electrons. And they're not actually two kind of like separate pools of molecules. So if you have a bunch of NAD plus, well, that's actually going to stimulate this. And so now you have to think about, okay, well, how would you have a lot of NAD plus? Well, if you're using the NAD in order to generate your lipid, you're going to be generating a lot of NAD plus, and then the NAD plus is going to stimulate this process. So hopefully this kind of helps show you the beauty of how these different pathways are interconnected and regulating one another. But sometimes just those allosteric regulators, the amount of various molecules aren't enough. And sometimes we actually need hormonal stimulation. This stimulation is often going to be indirect, but it can be through like phosphorylation cascades like this. So, for example, we can see that insulin and glucagon are going to inversely regulate our glycogenesis, our making of glycogen, our storage form of glucose, and our glycogenolysis, our breaking down of glycogen, give us glucose. Insulin is going to stimulate, basically, if we have a high blood sugar, insulin is going to be released. It's going to stimulate the uptake and storage of glucose as glycogen. And conversely, it's kind of like glucagon, the kind of reverse of the insulin, is going to do the opposite. It's going to stimulate the breakdown. So we can see that different directions of pathways are often going to be inhibited by the opposite enzyme. Once again, we see if we have a separate enzyme in each direction with a one-directional arrow for each, that's telling us that that enzyme catalyzes this so-called irreversible reaction. Whereas the reactions with double-headed arrows, these are going to reverse more easily reversible. So that was kind of activation by our hormones. Those are often going to be indirect. Sometimes what you might see either for that and or for the allosteric regulators, so those small molecules, is instead of showing them as like a green arrow or a green triangle or a red X, you'll see like an arrow, so often like a green arrow indicating a stimulation, a line with a kind of like a bar at the end, so like a horizontal line at the end that's indicating something's inhibiting or blocking. So there are different ways in which you could show that. Another thing that might be shown on a pathway is going to have multiple arrows in a row. This is kind of showing you that you have a multi-step reaction. So this isn't going directly from ribose 5 phosphate to nucleotides. It's actually multi-step. This takes us back to thinking about something else about the pentose phosphate pathway. We can see here that we're basically going from glucose 6 phosphate to ribulose 5 phosphate. Now, even if you don't know what ribulose 5 phosphate is, you see that we lost the CO2 here. So we have one less carbon here. When we lose a carbon, basically we're losing the opportunity to make energy later on. We can see that the pentose phosphate pathway didn't provide any energy, and we're losing the opportunity to pull energy from by when we lose the CO2. So we wouldn't want to go this way if we want energy. If we want energy, well, now we're going to go and we're actually going to take the molecules of glycolysis. We can see that glycolysis itself is also going to be regulated by the amount of energy that we have. If we have low energy, it's going to basically um, is going to stimulate the forward pathway of glycolysis so we can break down the glucose. If we have high energy, as indicated by if we have low AMP or high ATP, this is basically going to inhibit the reverse process. So in this way, these pathways are going to be regulated, and you can track all this by just looking at what regulates what. Often you'll see that that a pathway is going to be inhibited by a product of the pathway. So for example, this hexokinase step, it's inhibited by glucose 6-phosphate, which is the product. So this is going to be like our feedback inhibition. And then we'll also see examples of feed-forward inhibition or feed-forward stimulation, so that we can basically prepare these enzymes for what's coming. To review, the key things to keep in mind are whether or not the reactions are single directional, so one directional arrow, which would indicate that something is metabolically irreversible, or bidirectional, which indicates that a reaction is going to be readily reversible. Look at what's coming in and what's coming out. Keep a key eye out for things like our NADP plus and our NADPH, our NAD plus and our NADH, our ATP. When do we spend it? When do we get it? Keep track of this because it's going to be important for dictating what pathway we want to go when. For example, in glucose metabolism, we can see that we're using NAD plus and generating NADH. If we want to keep glycolysis going, we're going to need to keep replenishing that NAD plus. If we then look and see how we can regenerate the NAD plus, well, we can see that we can actually use the electron transport chain. We first use the shuttle to get the equivalence of the NAD, the electron equivalent into the mitochondria, and this is going to regenerate our NAD plus. What if we don't have oxygen? Well, if we don't have oxygen, we can see we need oxygen to do this. So if we don't have oxygen, well, now how else can we regenerate the NAD plus? If we look at our chart, we can see that we can regenerate the NAD plus if we look at this lactate dehydrogenase. Lactate dehydrogenase is able to regenerate the NAD plus, and now we can keep glycolysis going. We see that this step is going to be easily reversible. So in fact, in our liver, the lactate can be converted back into pyruvate, and now pyruvate can be used either to go back up through gluconeogenesis to make glucose, or it can actually be broken down in order to give us energy. All of this is going to be dictated by things like our energy availability and various hormones and things like this, as we can see by looking at the regulators of these different enzymes. The amounts of these different regulatory molecules are going to depend on what pathways are being used and when energy is being used and things like this. So it's all interconnected and it's all beautiful, but it can all look really complex. The key thing is not to let yourself get kind of anxious about starting. Just take one molecule at a time and follow it as it goes. Follow those arrows, follow what's regulating what. When you get to a branch point, think about when you might want to go which way and why. Focus on the carbons, focus on the arrows, and focus on the beauty.
So don't get overwhelmed. I know it looks overwhelming, but kind of keep the big picture in mind while focusing on tracking one molecule at a time.